Good morning. I am Dr. Marie-Odilei SOYER-GOBILLARD, Emeritus Researcher, living south of France. First, sorry for my French accent. Second, our work was realized in collaboration with Professor Charles Sultan, endocrinologist Philippe Courtet, psychiatrist, and Laura Gaspari, pediatrics. We have no competitive financial interest. However, I am a concerned mother, therefore a victim, scientist, emeritus researcher at the French National Center of Scientific Research and president of the Orange France Association, Stop for Artificial Hormone for Pregnancies. Everyone is familiar for the serious consequences of the death which was prescribed to millions of pregnant women throughout the world in 71 in USA and 77 in France. If its somatic effects as genital malformation, infertility, cancer on children exposed in utero have long been recognized, it is not the case for psychiatric disorders as severe psychosis, as schizophrenia, or bipolarity. At the end of the 90s, somewhere in France, an agricultural engineer made the observation that his 3 children were suffering from various psychic and somatic pathologies. The elders suffering from bilateral cryptorchidism, micropenis, infertility due to azoospermia, and schizotypal character. The second from anxiety, depression, and eating disorders, coupled with small uterus and ovaries. The third, suffering from schizophrenia and severe depression associated with suicide attempts. He observed also the relationship with the fact that in all 3 cases his wife received medical treatment consisting of a synthetic hormone cocktail, DES, ethinylestradiol, plus synthetic progestin delay during her 3 pregnancies after a previous miscarriage. He conducted research in the world literature on the subject, and he wanted to expose his observation at a meeting of patient families collecting observations on the genital malformation of girls exposed in utero to death. Alas, he was condemned, rejected, and disclaimed by doctors, mostly gynecologists and psychiatrists, as well as associative members. Discouraged, he died shortly afterwards. In order to continue his work and regroup the family concerned by the origin of the heavy psychiatric pathology of their children, and despite the taboo surrounding such disease as psychosis, we gathered several mothers concerned and created in 2002 the Association of Patients Orange. We elaborate a detailed questionnaire sent to death mother and to death daughter, prepared with researchers and doctors. The whole Orange cohort is now registered on the epidemiological portal of French health database INSERM, French National Institute for Medical Research. In a work published in 2016, we found in 1,002 prenatally death-exposed children included in the French Orange cohort a high prevalence of psychiatric disorders, schizophrenia, bipolarity, behavioral disorders, eating disorders, suicides, and numerous suicide attempts. In this work, we show that prenatal exposure to death and or to ethinylestradiol is associated with severe psychiatric disorders that appear in adolescence or post-adolescence. These disorders are accompanied or not by somatic disorders, genital malformation, and or by cancers. In 17, the epigenetic mechanism of estrogen effects on the brain of children exposed in utero have been partly elucidated from the Orange cohort by the team of Professor Krebs from Sainte Anne Hospital in Paris. They discovered a specific hypermethylation at the level of two genes implicated into neurodevelopment after taking peripheral blood from psychotic patients and using their unexposed eldest as a control. It was published in PLOS ONE in April 19. An hypermethylation of the ZFP57 gene was observed in the group of psychotic exposed children. This gene, located on chromosome 6, is expressed very early in development. It is a transcription regulator at the level of many genes affecting, in particular, the neurodevelopment. Hypermethylation of the ADAM-TS9 gene promoter. This gene is involved in the control of organ shape, particularly in the development and function of the uterus and reproductive organs, often abnormal after in utero exposure to death, as well as in the control of development of the CNS nervous central system and in several types of cancer. Synthetic progestins may also induce the same psychiatric disorders in children exposed in utero and also autistic spectrum disorder. We found in our cohort 46 families representing 62 exposed to children to progestin only. Prescribed progestin to pregnant women of our cohort were specially 17 alpha-hydroxyprogesterone caproate, a synthetic progestin, against total indication in 2000 in France, but reauthorized in 2011 for pregnant women. The main psychiatric disorders were schizophrenia, bipolar disorder, somatic disorder also. In 2000, In 2017, in the team of Professor Paul Yao in China, Tongren Hospital of Wuhan, from animal experimentation, exposure of pregnant rats with levonorgestrel during 21 days, and observation of 10-week-old pups, a behavior autism-like was observed. So they analyzed the limbic area of the brain after dissection, and by using molecular probes, they found an hypermethylation of the promoter of gene ER-beta, the receptor nuclear to estrogen, located in the amygdala zone of the brain. 1 year after, always in the lab of Professor Poliao, they realized an epidemiological study in the island of Hainan, China. A population was analyzed, and 235 children with autism were found. The antecedents discovered were in utero exposure with prescribed progestin to avoid miscarriage, conception and early pregnancy on the contraceptive pill with progestin or estroprogestin, and 100% of mothers had consumed seafood contaminated with estroprogestin. Breeders adding estrogen, progestogen to fish and shellfish food to prevent them from laying their eggs and weighing more for sale. They made also animal controls, maternal rats fed with seafood contaminated with progestin, autism-like behavior of pups were observed. Progestin are known to induce GABA receptor dysfunction activation before birth. It is likely that modification of the activity of the GABAergic system could contribute to schizophrenia, anxiety, and depression. Moreover, the promoter of the gene ER-beta is hypermethylated. Also, some progestins have been banned from the market, others are not. Our data demonstrated that caution should be taken with regard to the use of this progestin during pregnancy and even outside this period during contraception in particular. We have collaborated with Professor Poliao in writing of a chapter included in the book Factors Affecting Neurodevelopment. Transgenerational effect. This figure schematizes the transgenerational inheritance of epigenetic defect over 3 generations. A pregnant woman and a father, representing the first generation of parents, carry epigenetic information to the fetus F1 and to his germ cells F2 of 3rd generation. In 2010, American researcher Oreilly and collaborators analyzing a cohort of nurses known as the Nurse Health Study found girl deaths included with significant percentage with major depressive disorders. More recently, still at the psychiatric level in the same cohort, enrolled in the Nurse Health Study 2, ongoing study on 3 generation participants, their mother and their children born alive, Kurmuzoglu and collaborators concluded in JAMA Pediatrics that exposure to death was associated with multigenerational neurodevelopmental deficit, attention, hyperactivity disorders. We report now the impact of death prescribed to suppress lactation of the mother of a sibling of 11 children on 10 children born after such treatment and their progeny on second, third, and fourth generation, by focusing particularly on psychiatric disorders. Mother took death during 3 months after every delivery to stop lactation. Only the first child, a girl, used as control was unexposed. Herself and her descendants are devoid of disorders. As shown on the following pedigree tree, you can see on the first line the parents, second line the children, 11 children, third line grandchildren, and fourth line grandchildren with a problem on the fourth grandchildren. Also, selection bias may affect our data. They provided from a family-patient association the high incidence of psychiatric disorders associated with somatic in the estrogen and or progestin-exposed subject is quite striking. It should be noted that in all the cases, the intrafamilial controls elder without any exposition were deprived of disorder. The orange cohort of children exposed in utero to death or to alpha-exethanylestradiol during pregnancy represent an almost experimental model to evaluate the toxicity of this product. It is a real-world experimental group in a real-life condition. So this could be considered as a model study for other endocrine disruptors. In 2004, the Greek biologist Theo Colborn said, "The fetus can be protected from endocrine disruptors, whatever they are, only at the zero dose." Thank you for your attention.